Good morning, everyone, and welcome back. And sorry for the delay and the last-minute traffic reassignment. Okay, we're all here, so we'll begin as always in the name of the Father, the Son, and the Holy Spirit. Blessed Trinity, we love you, we adore you, and we thank you for your many blessings, for the gift of life, and for having us here again to continue to consider your teachings with regards to these bioethical issues at the beginning and the end of human life. We ask always your inspiration, your guidance, so that we may be also an inspiration to those who surround us as we take your teachings to those who surround us. We pray in a very special way for those who are in most need of your divine mercy at this time. In the name of Christ, our Lord. Amen. So today is the last lecture of the Beginning of Life series. And then we'll start on the end of life issues until the end of the semester. Okay. As always, any questions or comments from previous lectures, material? I have yet to address a little bit the email that Monica sent with regards to the use of human embryonic stem cells for research in products that are out there in the market, and the distinction between formal and material cooperation is very important to make that distinction. But it will come in well also in the end of life issues, so I'll wait until we have everyone on board to talk about that, especially when she's back in class, all right? All right, so today what I have is a few topics from here and there, really. They're not necessarily connected to each other, but these are all clustered under other manipulation of the human embryo or the fetus, for better or for worse, because hopefully I'll end with a positive note. On the better side is the fetal medicine that is being, bless you, fetal medicine is being practiced today. Very interesting, positive use of sophisticated technology, right? So these are the 5 little topics that we'll cover today. We'll take a break somewhere in the middle. And first, starting with 3-parent embryos, then human-animal chimeras, prenatal diagnosis, the growth of human organs in animal models, and finally, fetal medicine. So let's look at 3-parent embryos first. Where does this come from? Maybe you have seen it in the news the past couple of years. Did anyone remember seeing this in the news? A lot of this stuff is coming out of the UK because since in vitro fertilization started there first, they have a leg up on all the manipulation of human embryos, and they've moved forward on it. Basically, what the UK has is some government regulations at the federal level, but they have these commissions, these bioethics commissions, which essentially are run in a utilitarian fashion. What do I mean by that? This is important for the bioethical analysis, and I understand I'm kind of jumping to the bioethics here, but I want to remind all of you these principles that essentially we have 2 ways of doing bioethics today, either in a utilitarian way or in a principle way. Right? Utilitarian is, if it works, do it, basically. In other words, it comes down to the end justifying the means. If it's a good end, then whatever means is reasonable, will get to that good end, then you can move ahead. Of course, what they have is these bioethics commissions that are government appointed, and they'll be certainly pluralistic, right? So they'll have representation from a variety, from the scientific community and even from the religious community, just to see that there's consultation. But at the end of the day, since democracy rules in those commissions, then it's by consensus. And it may come down to a simple majority. The commission may be 5 people, typically it will be an odd number so that they don't get locked into a tie, right? And at least 3 or 4 of the 5 will be in favor of the thing, and the thing will move forward. Further background to utilitarian mentality precisely because the end justifies the means, then many times in the means, what do we have? We have on one side legalism, meaning if it's not illegal, then it has the perception of being ethical. All right, that's an extremely dangerous position to be in. And quite frankly, most people in society operate that way, whether they're conscious of it or not. All right, consider this. If it's not illegal, right, as long as it's legal, then it has the perception of being ethical. The challenge with that, of course, is that there are many things that are not illegal and are highly unethical. For example, I use the example of adultery. When was the last time we saw someone getting arrested for infidelity? Right? We even laugh at it because it's laughable, really. However, it used to be illegal. There used to be a law against infidelity in this country. And of course, with secularism, all that went out the window. So now people are unfaithful and they don't get arrested for it. So it's legal. And I'm sure we all agree that it's highly unethical, right? That kind of thing. So that's a very dangerous position to say, well, it's not illegal, then it's ethical. That is called legalism, right? And it puts an excessive burden on the law because the law cannot cover every aspect of human life. So the law is minimalist, but the ethical standards have to be really above and beyond the law. The other aspect of utilitarianism is what we call proportionalism. Proportionalism is the overriding principle. Proportionalism means if there seems to be a proportion, if there's a greater good, then the thing should move forward. And sometimes that is even down to a physicalism. In other words, the actual physical size. The embryo is evidently smaller than any of us who are already born. So the embryo is going to lose out, right? And the embryo doesn't look like us who are born, so the embryo is not fully human for proportionalists. See, that's the challenge with proportionalism, is that everything is relative. There is an evident internal contradiction to that. It's so obvious that people don't see it. When I say that everything is relative, that is an absolute statement. So that in itself is a contradiction. If everything is relative, then that statement should be most things are relative. Because if everything is relative, that's an absolute statement, which is a contradiction of everything being relative. So there are these internal philosophical contradictions in the utilitarian mentality that is totally glossed over because we have essentially forgotten how to think logically, and how to stick to certain values and principles, regardless of whether the outcome is positive or negative. So this proportionalism applies a lot on the issues of the beginning of human life. And what happens there is that essentially because the embryo is not fully developed, then the embryo is not fully human. The fallacy in that is that we're placing humanity on a developmental scale. And who of us is fully developed, right? We would say by the same argument, a newborn is not fully developed because a newborn cannot feed for himself or so forth. A child is not fully developed. A teenager is not fully developed. I know some adults who are not fully developed either, especially intellectually, right? Exactly. Sarcastically, I call them chronological adults, right? But then they don't act as adults. So really, at the end of the day, we're always in some stage of development. Where do we draw the line? If we're always in some stage of development, the line is going to be arbitrary, unless we draw it at the very beginning of that development, which is the zygote. So bottom line for utilitarian mentality, legalism and proportionalism gets people to justify just about any procedure as long as the outcome or the end is a good end. You see? And the means, like we say, by hook or crook, but move forward. And so they have these commissions that pretty much just to say that the government, just for the government to say that it went through some ethical review committee, right? Okay. So the alternative, since I'm on that, I might as well give the alternative, which is obviously what I espouse and what this whole program is based on, is what we call principled ethics based on certain fundamental principles. And those fundamental principles are absolute. In other words, they hold always, every time, everywhere, like the dignity and the sanctity of human life, from conception to natural death. That's a basic one. That's a fundamental one when we do these bioethical analysis. I know it's evident for you, but for many in secular society, that is not acceptable. See? You say, well, that's your opinion. So there we go with the relativism again. But principled ethics is based on certain fundamental principles that are intrinsically good and should always be upheld. And that will make certain procedures intrinsically evil because they will violate those fundamental principles. See? That's why we call it principled ethics. That is the Catholic ethics that is applied for the life issues and for other issues too. So bottom line is with principled ethics, we hold because there are some intrinsically good values and principles to be held, then violation of those principles is an intrinsic evil. And no amount of good intentions, no amount of good intentions will justify the means that are wrong or unethical means, all right? And so forever, these two ways of doing bioethics are going to be at odds with each other, the utilitarian mentality or the principle mentality. And that's where right now we are basically at a deadlock. And that is also why in principle bioethics, we use nature. We allow nature to tell us because we're looking for some objective fact that is irrespective of my beliefs or my values or my way of thinking. Nature happens in a way that is independent of whether we understand it or not, or like it or not. Nature was here before we got here. Nature continues, and hopefully will be here after we leave. And so nature has a way of doing things, and we look at nature to tell us what is proper and what is improper. You can see right away that that has a lot of application for the beginning of life issues, right? It also has a lot of application for the end of life issues. And now it also has a lot of application for the environmental issues. If we're going to be consistent with an argument of respect for human life and for non-human life. All right, so that's a bit of background. Now we go into the three-parent embryo, having said all that, and let's see how that works. First, stop for a moment. Questions or comments? You see these two mindsets, they're kind of locked in place. And of course, they also define even political systems and how political systems move forward, right? Because then it gets to the level of ideology. And when those political systems lock into those ideologies, that becomes then the political propaganda or the machinery for re-election and so forth.
That's the mistake that we make, that you said, Father, because it's legal.
Right.
They think that it's right.
Exactly.
And why, justice, a lot of time, it's a lot of negotiations, really.
It is. The power of the argument, right? Exactly. And the compromise, and it's the art. When I remember my brother teaches political science, and he was telling me one practical definition of politics is the art of the possible here and now. The art of the possible here and now. Okay, so no politics is going to be perfect, really. And particularly, that's why this is the American experiment, is this democracy, right? Where we have to negotiate, we have to enter into the dialogue, and we have to come to some agreement. And it's going to be by consensus because we are 325 million people in the United States. And not everyone is ever going to agree on any one issue, even the way we like the coffee or whatever, or which is the best coffee. With or without sugar, or salt and pepper, So let alone the vital issues of human life. So that's the challenge. And that's why even when something is legal, doesn't mean necessarily that it's ethical. We have to go to a higher standard, which is the conscience. And I also like very much that for us in Catholicism, we follow that pathway of the law, the positive law that we call, right? The civil laws that are in the books, including traffic laws, everything, those are what we call positive or civil laws made by man, made by the human, typically in the legislative branch, are supposed to be a reflection of natural law, right? Why is it a civil law? You know, why is murder wrong civilly? Why is there a law against murder? Because human life is precious. And so natural law is the background to good civil law. And the background to natural law is divine law, because nature ultimately is created one way or another by the Creator in the laws that God established for this planet to have life on it. And I've mentioned this before also, all the founding fathers of the United States, when this American experiment was congealing in the Continental Congress and the writing of the Constitution and the Bill of Rights and all that, they were all natural law philosophers. Back then, that was the standard model for Europe. They followed natural law, okay? Many of them were also Masons and everything else. That was a cultural thing, but philosophically, they were natural law advocates. And so you see that many of the laws of this country, including the Constitution, the US Constitution, have those principles that include God. In God we trust, right? And the delegation of power from God to the people, and then the people delegate that power and authority to the representatives legitimately elected and so forth. Very different model from, let's say, a tribal hierarchy where power is absolute and you have these dynasties and it's inherited and so forth. Very, very different model. It's a power that flows from the people, from God to the people, and the people, from the people to the legitimate authorities elected and so forth. So there's a system, there's an organization. That order is reflecting what happens in nature, natural order. Right? A lot of that has been ditched in today's society, secularism, including the European Constitution, when the European Union was formed. And I remember St. John Paul II being very, very upset at the, what was equivalent to the Continental Congress, what happened in Europe when they were putting together their constitution for the Union, right? Where they explicitly excluded God or any reference to the Almighty or to eternal life or any reference to the supernatural, totally excluded on purpose from the European Constitution, right? Totally secular. And then the irony is that they elect the hymn for the European Union, right, is Beethoven's Fifth Symphony, which has all kinds of religious connotations in it. And it's a hymn that is even used in church. Right? The Beethoven's Fifth Symphony. So go figure. All right, 3-parent embryos. Let's get to it. Have you heard of mitochondrial diseases? Mitochondrial diseases. We have to put in the plural because it's a cluster of diseases, and it has to do with the mitochondria. And let me introduce you to the mitochondria, or remind you of it from our high school biology. Here is the idealized cell. It doesn't really exist in nature, okay, but this is an idealized cell that is showing all of the organelles inside the cell. It's a good little setup also in anticipation for the next course in the summertime that will be environmental bioethics. We're going to do a little review of biology there, just basic biology, right? But in anticipation to that, here's a review of the cell, which is the basic unit of life, organically speaking, right? The cell is the basic unit of life, organically speaking. Of course, we know that we have to put the cell in context also, because if I just have one cell sitting here on top of my palm, that's not going to function properly, right? That cell needs to be connected to other cells and act in collaboration for tissues, organs, and systems, et cetera, et cetera. But what happens? That the cell, in order to function properly, which is what we call metabolism at the cellular level, it's just body function or cellular function, right? It's life at the organic level, at the level of the cell, is metabolism. So what does that metabolism, that proper function, is all these little structures that are called organelles. So just as our bodies have organs to function properly, for the whole body to function properly, the cell as a unit in itself also has these little organs inside, these little structures that are called organelles, right? Organelles. So the big organelle in somewhere around the center of the cell generally is the nucleus. And the main function of that nucleus is to hold the DNA and some RNA and proteins, of course, in an organized way, and it is protected. It's protected. So the DNA is stored in the cell within the nucleus. Then that message is sent out, as we saw in the central dogma. The messenger RNA is sent out to the cytoplasm, specifically to other little organelles that are called ribosomes. And in the ribosomes, which are little structures, it's a complex of RNA and proteins, they are the ones who decipher the messenger RNA code, and they make the proteins. So protein synthesis occurs in the ribosomes, which is out in the cytoplasm. The cytoplasm is the liquid aspect of the cell, and it is mostly water, anywhere from 80 to 90% water. And that is the soup, if you will, where all the cellular activity is occurring, the metabolism. There are other organelles. One that I'm focusing on today is the mitochondrion. Mitochondrion, one mitochondrion, several mitochondria. All right, let me see if I can make a little bigger here. Okay, you read it there? Okay, so the mitochondrion is an organelle that is responsible for recharging ATPs. Recharging ATPs. The ATPs are the energy molecules of the cell. Excuse me. The energy molecule of the cell. And ATP is an abbreviation for adenosine triphosphate. You don't have to worry about so much detail, but just to know that the adenosine triphosphate is an organic molecule that is in charge of supplying energy to the cell for metabolism, for body function. Just like the car needs energy to move, to function, right? The cell also needs energy. What is the energy molecule of the car? Gasoline, right? Gasoline. So the energy molecule of the cell is the ATP, and this ATP gets recharged at the mitochondrion, right? One mitochondrion, several mitochondria. That's just Latin. So it's a vital organ, a vital organelle of the cell. And we have several thousand mitochondria in each cell of our body. Here's an actual electron micrograph. You see it's a double membrane, and it has the scriptae that go in and out. That's where the actual photosynthesis occurs. You can also see in this micrograph the endoplasmic reticulum, which is this folding, extending out from the nuclear membrane, and the little dots are the ribosomes. You can see them lined up here, so it's very interesting. This is an actual photograph, and it's getting down almost to the macromolecular level. I had several thousand. I should have put the amplification, but I don't have it. This is several thousand times magnification. So some people have what is known as mitochondrial disease for a variety of reasons. These mitochondria are defective, they may not be in sufficient number, or may be damaged so that they don't function properly, because they are in charge essentially of the energy of the cell, and organs are made up of tissues and tissues are made up of cells, right? So this will impact the organs of the body and the whole metabolism of our body. What is your expectation when these mitochondria are few in number or defective in structure and function? You expect a general weakness of the person. And that weakness can be chronic, and that weakness can be so debilitating that it will actually impair them from normal functioning. So it's a serious disease. It's not necessarily lethal, but it's significant. So the other thing to know about mitochondria is they also have their own DNA. That's why we were able to talk about mitochondrial Eve, right?
Yeah.
Remember mitochondrial Eve from last course? This is how it works. So we all have mitochondria in every cell of our body, correct? In order for that cell to be alive and function. But when we take it to the first individual, what is the first stage of our individual beginning? The zygote, right? It's so obvious, you should know this in your sleep. So when we get to the zygote, we're talking about a single cell. And we were all zygotes at one point, a while back. That's a single cell. The cytoplasm of that zygote, the cytoplasm of the zygote, did it come from the egg or the sperm? Which has more cytoplasm, the egg or the sperm? Which got fertilized? The egg got fertilized. The sperm fertilized the egg. But the sperm is mostly a nucleus with a nuclear cap, you know, with a membrane, right? With a cell membrane and a little bit of cytoplasm. But the sperm has very little cytoplasm, which, by the way, is poured into the egg at fertilization. That's in all mammals. But we can say that the vast majority of the cytoplasm in the zygote comes from the egg, meaning from the mother, meaning maternally inherited. So all cytoplasm of all zygotes, of all mammals, is maternally inherited. 99.999% of the volume of that cytoplasm comes from the mother, right? And therefore, so do the mitochondria, because they're in the cytoplasm. Now, I remember reading an article way back years ago from the New England Journal of Medicine that some researchers had actually found a few, talking maybe a dozen or so, male mitochondria in a zygote, okay? It's okay because, remember, the sperm also needs to travel, right? That the flagellum is wiggling very fast, so that's consuming energy, a lot of energy. And it needs the flagellum, especially the head of the flagellum, the proximal end where it's attached to the rest of the body of the sperm, that needs a lot of energy. So there's cytoplasm around that structure there. Mm-hmm. And there's not a cytoplasm, there are mitochondria that are supplying the energy for the sperm to be motile. And those mitochondria get poured into the egg together with the male pronucleus, right? But they're very few in number, number one. Number two, the egg cytoplasm attacks those mitochondria as foreign bodies and destroys them. So at the end of the day, you have very few, if any, male mitochondria left in the zygote. And therefore, in the new individual, because from the zygote will come every cell of our body, right? From the zygote will come every cell of our body through mitosis, forever mitosis, the millions and millions of mitosis that makes up now the trillions of cells that we have. So you see the progress? And every time these mitochondria are coming from that maternal inheritance, from that maternal line. Yes?
Only with the mitochondria, or with the other organelles?
All the other organelles, okay? It's just that the mitochondria, one characteristic of the mitochondria is that they have their own DNA also. They have their own DNA, and that's the mitochondrial DNA. And that's the one that was traced to find out mitochondrial Eve, right? The first female human that started all the human line. all the human species, the Homo sapiens line, right? Which is the only Homo living today. And so it's maternally inherited. It has its own DNA, which is not nuclear DNA. It's a small DNA, and the vast majority of that DNA codes for the mitochondrial structures. And that's also why it's believed that evolutionarily, this mitochondrion was originally a microorganism that got sucked into the cell, but was not fully destroyed through phagocytosis, right? Survived the attack and kept its own DNA, and eventually the only thing that survived is the actual energy producing characteristic of it. We're talking way back in evolutionary time, okay? Because we find that all,
Oh, thank you.
you're welcome, that all multicellular organisms, right, have mitochondria in them. So this happened at the very beginning of evolution. In addition to the mitochondria, for plant cells, they seem to have also trapped another microorganism that was a photosynthetic organism, so probably some cyanobacteria-like structure or organism that got sucked in and survived the attack. And that gave rise eventually to the chloroplast, which plants have and we don't have. And what happens with the chloroplast? That's where photosynthesis occurs. The photosynthetic pigments are inside the chloroplast, which is another organelle in plant cells that we don't have because we don't photosynthesize, but plants do have. So plants have, in addition to mitochondria, they also have chloroplasts. And chloroplasts also have their own DNA. So one could potentially do the same analysis on the chloroplast and find out the original plants where they came from, looking at the chloroplastic DNA, you know, and tracing it back too. It's not our thing because it doesn't have to do with human life. But just to mention that there's another one of these organelles in the plant system. Imagine that it was originally most likely another organism, independent, that got sucked in and not digested, and has given rise to the producers of the world, to all of the plants and the possibility of plants today being able to photosynthesize and therefore maintain life on Earth, right? Because they are producers of glucose precisely, which is the fuel. That's what the mitochondria burns to obtain the energy, is burning glucose. All right, anyway, all this to point out that mitochondria are essential in the cells. When they're few in number or dysfunctional, that's going to cause a general weakness of the person to the point that it can be so debilitating the person is not functioning properly. So, because it's in the cytoplasm of the egg, right, the mitochondrial disease will affect women more than men in general, but will be also passed on by women, by the mother, because of the egg. Because of the cytoplasm in the egg, the mitochondria in the cytoplasm of the egg, and not the sperm. Okay, so in a utilitarian way, how to bypass this. Before we look at bypass, one last thing. Let's assume for a moment that this cell here is actually an egg, an ovum, all right? An ovum, so that means it's a gamete, that means it's haploid, and essentially the only thing we would have to change here or just note is that this DNA is not diploid. They're not chromosome pairs. There's a single set of chromosomes, just haploid. 23 chromosomes for the human instead of 23 pairs. If we make essentially that change, this cell becomes a gamete, it's haploid, becomes an egg, all right? All right, so let's assume that this is an egg, and then this becomes the pronucleus, and is the DNA, is the nucleus defective? What is defective in this mitochondrial diseased egg? The mitochondria. Is the nucleus defective? No, it's not a nuclear disease, it's a mitochondrial disease. How about the ribosomes? Are the ribosomes effective? It's not a ribosomal disease. Okay, the Golgi apparatus, which is in charge of getting rid of waste, the endoplasmic reticulum, the centriole has to do with the cell division, with mitosis, all the other, the lysosomes, all the other organelles are functional. It's only the mitochondria. Particularly for reproduction or for fertilization or for generating a new human being, what is important of the egg is the nucleus of the egg, the pronucleus, because that has the genetic material that will generate a new individual in fertilization, in pairing up, infusing with the male pronucleus, right, with the sperm pronucleus. So what is defective, if you will, is the cytoplasmic fraction of the cell, not the nuclear fraction. So the nucleus is good, all right? Okay, giving all that background, now, this is what these scientists came up with to bypass mitochondrial diseases. In other words, to essentially ditch the defective cytoplasm, or to save the healthy or intact nucleus of the egg. There are two ways of doing it, basically either before or after fertilization. So let's look at model 1. The repair occurs at the level of the embryo. In model 2, the repair occurs at the level of the egg. Here we have the mother who wants to have a child. Here's the father, potential father who wants to have a child. Let's assume that these people are married, okay? So these are husband and wife. We need another woman, a second woman who does not have mitochondrial disease. So this woman, the wife, the potential mother, has mitochondrial disease. What we don't want from her is her defective mitochondria, meaning we don't want her defective cytoplasm. But the nucleus is good. Okay, so this is what happens. There is fertilization, of course. Where is the fertilization going to occur? Because we're going to manipulate this zygote now. It's in vitro. This needs in vitro necessarily. There's no way to do this in the natural and then go in there and try to extract the early embryo or even the zygote. Impossible, okay? So this presupposes in vitro, absolutely, has to be done, manipulated in the lab. Okay, so in the lab, in vitro, the woman's egg is fertilized by the sperm of her husband. Now we have a zygote with mitochondrial disease, right? Because this is her whole entire egg was fertilized. That same father, that same man, fertilizes the egg of the donor woman. Okay? You already see big bioethical issues here. Essentially, we have cellular infidelity going on. We have cellular adultery. Even without the in vitro infidelity, without even the pleasure of it. Okay? Exactly. So, yeah. To the tune of several $1,000s, maybe $20,000, $30,000, $40,000. So go figure. Yeah, it's called arrogance, right? All right, but here we go. So this donor woman's egg is fertilized by the husband of the other lady. But we're not interested, what are we interested in? Her mitochondria, her cytoplasm. So now we ditch out the nucleus, which is already a diploid nucleus, which is already the nucleus of a zygote. So we ditch out the very core of that new human being. Instantly, when we do that, we kill that human being, right? Because it's like taking out the heart and the brain of the new human being, because we're getting rid of the DNA that codes for the new individual. So here's the first killing, here's the first murder right here. We ditch the nucleus, we enucleate the zygote. Now we have an enucleated zygote. We have a shell, if you will, that is healthy. Organically speaking, the mitochondria are healthy, intact. What do we do on the other side? Well, we take the diseased zygote, in other words, the mitochondrial disease zygote, and we're only interested in the nucleus of that zygote, the diploid nucleus of that zygote, which in principle is healthy because it's only the mitochondria out in the cytoplasm that is diseased, right? So we suck out with these little tiny cannulas, glass pipettes, suck out that nucleus and insert it into the enucleated zygote of the other woman. And now we have a new zygote that has the nucleus of the husband and wife and has the cytoplasm of the donor. And that's why it's 3 parents. Literally, organically, 3 parents. It's not even like the cloning, like the reproductive cloning, where you have, the cytoplasm of one and then the nucleus of the other, but the diploid nucleus is already inserted directly. So that's why this child truly has 3 parents, okay? As half the nucleus comes from the mom, the other half of the nucleus comes from the dad, and the cytoplasm comes from the donor. Yep.
So then, like,
when? Okay.
Well, that's the thing. Like, are you saying then that technically 2 babies are killed to then create a new one?
Right. Okay. So let's back it up for a moment. When would ensoulment happen with our analysis, with our right at syngamy, right?
Right.
Syngamy is here and syngamy is here. So according to our model, this zygote would have a soul. It's an ensouled zygote. It's a human zygote with mitochondrial disease, just like his mother or her mother. This could be a male or a female. His mother is mitochondrial disease. His mother used to be a zygote many years ago and has a soul, right? We assume that the mother is not soulless in the philosophical way. How about the donor, the infidelity zygote, the victim of infidelity, also has a soul, right? And is a victim, so she's not to blame or he's not to blame for the infidelity. He's a victim of that infidelity, but he has a soul, we assume, in our model of logical consistency at completion of syngamy. Bang. Now here, what happened to this soul? This soul, this human was killed, and this soul went to the mercy of God to pray for these people for all eternity. All right? So this soul has gone to God, just like any zygote dying. All right? How about here?
Still alive.
Yes.
Alive, right?
Yeah, in an argument of logical consistency, of course, this is a model that we're proposing philosophically and theologically. The soul is within that nucleus, kind of parallel to a human body that starts getting chopped up, right?
Because that's genetic material, right?
Exactly.
It's like you kill a child to put an organ in another one.
Right.
Yes, but what organ? We're putting not only a vital organ, but we're putting the most vital organ, the only vital organ at this point that is left, which is the nucleus.
Because the mitochondria is not as important, obviously, as the organ.
Exactly.
That's where I wasn't sure.
Yes.
It's like the soul is in the nucleus.
Exactly. Analogously, what would we say a functional analog of this to our human body, to our born human body? The soul would reside minimally in the brain, not the heart. The heart, organically, the heart is a muscle.
Right.
Cannot think, cannot feel, cannot pass judgment. We speak analogically or symbolically, we speak about the heart, the seat of conscience, or the gut. Even for the Jewish people, it was the gut, the entrails. But really, rationally, it has to be the brain, because that's where we make the rational decisions, even about good or evil. So the conscience needs to reside in the brain. And then think about it. We can cut off, or heaven forbid, we don't want to cut off limbs of people, but let's say from a blast, from a landmine or something like that, we know people who are paraplegic, we know people who are quadriplegic. Functionally, it's the same as if they didn't have their hands, their legs or arms. They just have the trunk and the head. That's still a human being. Does that human being have a soul? Absolutely. And the trunk, what is the trunk doing? The trunk is the vital system that is the support system, the organic support system for the brain.
Like the cytoplasm.
Yes, exactly. Analogically, and that's why we call the structures inside the cytoplasm organelles, because they are analogous to the organs of the body. So these are microscopic organs, if you will, but functionally they're the same. So the trunk, the organs in the trunk are a support system, or the vital support system for the head, for the brain, and not the other way around. It's not that the brain is here to maintain the trunk alive, right? Very important to be clear on that, because that has philosophical and theological implications. We would have to assume then, in an argument of logical consistency, that the soul is still hanging on for dear life inside that nucleus, with the fate of what's going to happen when that nucleus is put into the new cytoplasm, into the new shell. It's like, I remember when I was studying in Rome, my thesis, my dissertation director, actually, Dr. Demmer, Klaus Demmer, wrote an article on head transplants. And right, and his point was strictly a theological and philosophical and an anthropology argument, anthropological, that it's not really a head transplant, it's a body transplant. What is important about the transplant is not the body, it's the head. So it's a body transplant. You know, just like when we do a kidney transplant, we don't say that that's a body. It's not that the body is transplanted into the kidney, it's that the kidney is transplanted into the body. So the same. It's not that the head is transplanted onto another body, it's that another body is transplanted onto the head. Yeah? So it's a body transplant. It's not a head transplant. Even though the head physically is smaller than the body. So, right, so just to set things right, and where's the priority, in other words? And where does that conscience reside precisely, which is in the head. So here, the nucleus. All right, so that's model number one. Model number two. This is the repair at the level of the egg. What do they do? So the swap, the nuclear swap, essentially here, the nuclear swap between eggs happens before fertilization. Okay? So technically, there's no real infidelity here. You think about it. Okay? Unless we consider the cytoplasm, cytoplasmic infidelity. Because here we have the mother's haploid nucleus, and notice how it's represented by half the orange, all right? Meaning that it's a haploid nucleus, which is true. And the donor's haploid nucleus. And then there's a nuclear swap is done just like remember in somatic cell nuclear transfer, in cloning, there's a nuclear swap, right? We enucleate the cytoplasm, the egg of the donor, because we're looking for that healthy cytoplasm. And we capture, retrieve the nucleus of the mother, the potential mother, thus discarding the diseased cytoplasm. And we take that haploid nucleus, the egg nucleus, insert it into the shell cytoplasm, the healthy shell, right? Now we have, in principle, a healthy egg to be fertilized. Still have to be done in vitro, but the fertilization happens once there is that reconstructed, reconstituted healthy egg, meaning the pronucleus of the mother and the shell, the cytoplasm of the donor, of the healthy donor. So functionally, by this technique, one has also gotten rid of the defective mitochondria. Fertilization happens, hopefully. We have to see after how many trials, right? Can't expect this to happen at the first shot. We know that from cloning mammals, the average is about 250 trials, right? For it to work. And then, again, the idea is to grow that zygote to the blastocyst stage, 3 to 5 days in the incubator at body temperature, and then insert. Bam, the same principles apply, the same statistical values apply as in IVF. On average, 3 to 4 embryos will be inserted, 3 or 4 blastocysts, right, will be inserted on the hope that one will implant and so on and so forth. And everything else is also available for that mother, for those parents, including the pre-implantation diagnosis and all the genetic, which you can see here. Of course, why not? Because that's available. You can see that they're going to do a lot of genetic testing on this embryo, simply because they want to make sure that that mitochondria is indeed healthy, that it's the proper egg, and so forth. So they're going to do the genetic testing. This is highly manipulative. So we can expect all the other issues that I mentioned, all the evil issues that I mentioned in IVF to kick in here also, and then some, because in addition to the gross manipulation that has been done here by really forcing this pregnancy to take place, right? Has any one of these people, the people involved, the clinicians, the technicians, and the society who has approved this, and the boards, the ethical boards that approve, any of these people have considered this little guy in here, that he may actually want to have 3 parents or not? Okay? So anthropologically and psychologically, when this human being grows and wants to know who is his mother or her mother, okay? And the whole manipulation that went on, oh no, but it was to make you healthy. Yes, but in the meantime, you have manipulated me to such an extent. And what about my siblings who died so that I could be alive today, who did not die, were killed so that I could exist? Is it going to be to the tune that 200 of my siblings were killed so that I could be around today, to satisfy your absolute desire for control over manufacturing me, right? So it's just too much. I mean, it's totally, this is the craziness that kicks in when we put God out of the equation of his most fundamental characteristic, which is creator. And what can be more fundamental than creator of the human life that he desires or not desires in humanity? Okay, Marina?
It's not right, obviously. But if they, it has to be like only one cycle, that of course they don't do it with only one.
Right.
In this case, it will be acceptable or not? Because if the egg is not considered like a cycle, right? Like a human being.
Correct. The egg, an unfertilized egg, is haploid and it's an extension of the ovary follicles. It's a cell of the mother. It's a cell of the woman's body, just like the sperm are cells of the man. So they're not individuals in themselves, right? Just like skin cells or liver cells or bone cells.
The issue is the manipulation that is in vitro. And all the ones that are killed,
exactly.
All the ones that are killed is in
The one on the right is not nearly as bad as the right left.
Like the trial. You have to do it.
Yeah. So there are fewer offenses, if you will, on this model than on this model, because at least there is not that cellular infidelity occurring with the fertilization of 2 eggs and the direct killing of 1. But the whole procedure is tainted, all right? And I'll tell you, in a sense, it's really, at the end of the day, it's quite simple for us. It's tainted because it's out of place. In other words, it's extremely indicative in vitro, that adjective. All right, so we have adjective and we have noun. Remember, the noun is a substantive, which means the substance, the quid est, what is happening. What is happening is fertilization. We talk about a human fertilization, which is the beginning of a new human being, right? So that fertilization either happens in vivo or happens in vitro. And in this case, that adjective that qualifies the fertilization is the bioethical analysis in itself. We just don't have the right to take it out of place, to do it in a lab, because when we do it in a lab, now we are manufacturing a human being. We're manufacturing. Exactly, it's contra natura. That's why we use nature, the locus. That's why I place so much emphasis on that ampulla. If you see my slides from the undergraduate course, practically never lecture, I have that standard slide of where fertilization occurs in the ampulla. And I repeat that through the whole course, and you wouldn't believe how many don't get it still. As physical, as physiological as it is, totally independent of our belief, our understanding, whether we like it or not, that's where it has occurred for millions of years, because that's where the good Lord wants it for all of the reasons that we know. The selective pressure and all that, there's a wisdom in that design that is just absolutely elegant. And we need to leave it alone. We just need to leave it alone. Whenever we start manipulating, we're risking the life of these human beings. And they start becoming products that are being manufactured. And then, of course, with products, we're going to have a quality control because we want the best product. I'm paying $20,000, $30,000, $50,000. I want the best possible product. And so the defectives, discarded. Okay? That is secular. That is the utilitarian mentality. But Father, this is all about a new life for this couple that is being wrenched out of the will of God. That's what it is. Yes, it is. But finish the sentence. You want to do it, come hell or high water, because I want it and I can pay for it. And the technique is available. So you see, it can be done, but it may not be done. It should not be done. That's the bottom line.
Is utilitarianism the same as moral relativism?
Yes, it's a type of moral relativism because everything is relative. You know, there are no absolutes. There are no absolutes. The absolutes are, it's your opinion. And you're entitled to it, but I have mine. And
what benefits you at the moment?
Sorry?
What benefits that person at the moment?
Yes, exactly.
Something that benefits them like today might not benefit them tomorrow, and they say that it's no longer.
It's no longer. So it's relative, you see. It's circumstantial. It's based on the circumstances. If I can pay for it, if the technology can take me there now, because this technology was not around earlier. But now we've gotten that sophisticated that it can be done. It can. So bioethics today is driven a lot of technology. But you can see the links because when we talk about technology and technological advancement, those are the high-tech startups that are high risk but high potential for high return. So when we talk about technology, we talk about economy. And when we talk about economy, we talk about politics. And then it goes into, it has the potential for going into ideology. And then the spin, and it becomes partisan, and that's where we get locked in, right? Yeah.
Father, in a cellular level, speaking, does this donor have to be like a relative? I mean, it has to be like, because when we saw Dolly, it was the same individual.
Right.
To make this the nuclear swap. In this
Right.
way, it can be like any donor.
Exactly.
Or it have to be like a compatibility between the cells.
Exactly. So keep in mind, when you go here, you're going into the in vitro model. All right. So in vitro has catalogs. For decades now, human sperm has been frozen, cryopreserved. And more recently, human eggs now can also be cryopreserved. If they can be cryopreserved, they can be stored, they can be shipped, and they can be cataloged and bought and sold. Which means that the donor obviously is a person different from the mother. Okay? I'm sure she's being compensated for her donation. The father is the father of the kid, but doesn't have to be the husband of the mother. Can be another donor too that has potent sperm, and that is happening now quite a bit in vitro clinics throughout the United States and throughout the world, that some men happen to have very potent sperm. Well, they're continuously masturbating and selling their sperm to these in vitro clinics throughout the US and throughout the world because their sperm is very potent, and therefore they are really the father of many, who knows, hundreds of kids from many different women who don't even know who they are, okay? But it's out there. So you can see that any possible combination applies.
Okay, so it doesn't have like a compatibility between at the cellular level, no.
Exactly.
It can be just, I mean, the shell doesn't have to be like at least similar to the mother, no.
How similar?
It can be anyone.
Right.
Okay.
So what's the bottom line here? Homo sapiens, they have to belong to the same species.
That's the,
That's about it.
There's not going to be a rejection
There will be.
Okay.
That's what I'm saying. But it's trial and error. You know, how do they obtain the egg of this mother? You think they went out and took out one egg when she was ovulating? No, hyperovulation, dozen of eggs. All right, 1, 2, 3 dozen eggs. So it works with excess. How do they obtain the egg of the donor? Again, you think they're going to wait for her ovulation to take out one egg? Again, hyperovulation over there. So again, you got dozens of these eggs. They can be frozen, so it's just excess material to start with.
Oh, then maybe they try with multiple donors?
Absolutely. Whatever works, that's the whole point. If it works, do it. That's the whole point. It's difficult to go there for us because we're not used to justifying anything so long as at some point she walks out pregnant, or she can rent a womb also if she also want to get pregnant, right? As long as she has baby in hand at some point, whatever works. She could be single, she could be married to another woman, she could be married to 3 men, who knows? At some point, you know, polygamy has to be justified because it doesn't have to be between 1 man and 1 woman. It doesn't have to be between 2.
I know the bioethical is wrong, but I was trying to understand like the biology.
Yeah, the biology is just,
I mean,
the biology is trial and error. They don't say that this is a simplified, this is the one that worked, okay?
yeah, exactly.
But this is a simplified model. They don't tell you, just like with Dolly, they don't tell you that it took 258 trials. But it's there when you read the original paper, because he has to report in materials and methods how many attempts. See? So this is a simplified model, but the background to it, this is hyperovulation, this is hyperovulation, this is masturbation, etc., etc., etc. Okay? Yeah, it's there. And that's why we get into it, because these models look so nice. Here is a mother, here's a woman who has mitochondrial diseases, and now she has a healthy baby. Okay, so it looks so good, right? Yeah, it looks so good. And we can do it today. And by the way, it's very profitable. And so people can pay it, so why not? Okay, so you see, why not?
When do they do that at the reproductive center in the chapter?
I don't know here they've gotten this far yet. I know it's been done in UK. This came out, you know, the papers are there and so forth. This came out 2-3 years ago, a couple of years ago. And of course, yeah, it's recent. It's recent. But it's consistent with the industry, if you will, just offering more services that can be done, and they keep pushing the envelope, right? So why not? It's a nuclear swap, essentially. It's kind of, when you see it, it's an extension of cloning. It's an extension of cloning. It's just being creative in cloning by the procedure. So it can also be seen from the other perspective, no? Of the woman who has, let's say that this woman has good cytoplasm but has defective DNA. Then she could negotiate for the healthy DNA, the healthy nucleus of another woman. So it can work both ways. Yeah. Again, just keep in mind, it can be done, and the technological imperative. If it can be done, it will be done sooner or later. That's when, that's what utilitarian ethics allows us to do. So along those lines, not to depress you too much, but if it can be done with a human, why not with the non-human? Exactly. Here we go. Now here we bring it back to the good old USA. So human-animal chimeras. This came on the radar 3 years now, in August of 2016. NIH, which is the National Institutes of Health, as I mentioned, the 2 main branches of federal government that do research in the United States, that fund research to the tune of millions of dollars and billions of dollars, all right, every year, with our tax money, are the NIH for human research and NSF, National Science Foundation, for non-human. And of course, United States research is the leading research in the world by volume, by money, by publications, et cetera, all right? Even though there's a lot of significant research going on in other countries of the world, the sheer volume of the millions of people that we have in the US and the thousands and thousands of labs that are involved in the research, just by the volume, it is the largest human and non-human research occurring throughout the world. is in the United States. So in August of 2016, NIH was proposing to rescind or to relax a moratorium, The moratorium is a ban for using federal money, for using NIH money, right, on certain types of human research. For example, just to give you a very simple and obvious example, it is prohibited to do human research without informed consent. As simple as that. And if they're minors, then it has to be a proxy consent of the legitimate authority of that child, which would be typically the parents or the guardian, which could even be the state, and so on and so forth. But the regulations for consent, for informed consent, all right? That's an obvious one, and it's there. But there are many other moratoria, if you will. There are bans on research when we talk about humans, for example, torture and things like that. That would be obvious. All right. So there is a ban currently or there was back then, to generate in the lab, to manufacture chimeras. A chimera is a mixed species animal. It's an animal or a plant that is a hybrid of several species, okay? And the mythological chimeras is the goat that walks around with a human body and the goat head and so forth, which was also used as a symbol of Satan in the devil worship. But those are chimeras. They're mythological animals that occur in the minds of people.
And the symbol of the Sagittarius, right?
Exactly, yes. The unicorn, the Sagittarius, they're Centauro, right? They're centaurs. Most of them come from Greek mythology, but there are other mythologies. You see sometimes the Hindu mythology also has that, the elephant with a human head and so forth. They have these mixture mythological figures. The Egyptians also had it, but they're typically related to religion or mythology and that kind of thing. You know, they don't actually occur in nature because we know that species is specific. All right, but now it turns out we can do it in the lab to start generating these chimeras, particularly between human and non-human. And so there were two proposals. One was to relax the ban on generating human germlines in an animal. What is a germline? The germline is the precursor to the sexual organs. The germline is the gonads that will differentiate into ovaries for women or testicles for men. Okay? There's currently a ban on basically generating sexual organs in an animal, human sexual organs in an animal. What was the purpose for that? Well, the purpose for that obviously was to obtain, they weren't so much interested in human testicles in animals, they were interested in human ovaries in animals. Why? To obtain human eggs from an animal, from a mouse or a rabbit or a cow, so that they don't have to be hyper-ovulating women and risk the legal sues of burst ovaries and so on and so forth, and that's the hemorrhages, and also the good desire of not harming women with hyperovulation and the whole hassle and the risk, the health risks that is. Okay, so bottom line, that one to generate human germ lines in animals was specifically to generate human ovaries in animals. In other words, a ready source in the lab of human eggs without having to bother with them. Right? Okay, so you see that the ends are very good, the means are very evil. But this one that concerns us is a different line. It is to generate a substantially human brain in an animal. Yes, I know it sounds bizarre, but here is the actual text, okay? So I'm not kidding you, folks.
Why a person would want to do that?
Exactly, because... Exactly.
It's like giving a soul.
For us who are believers, okay, and even for non-believers who don't accept that they have a soul, they still have a soul in spite of themselves. And so that's the whole thing. Why? Well, there has to be a motive, right? And the motive is very good. The motive is called the Human Brain Project. Remember the Human Genome Project in the 90s? I remember it. It was supposed to take 10 years. And then they locked into politics. And finally, between Craig Venter and the other fellow who's the head of NIH now, anyway, took it to term. And eventually, it was about 2 years late, but finally, the human genome was deciphered, right? The 3 billion base pairs, which I believe they only use about 100 subjects, human subjects that represent the whole population of the world, the 7 billion it was. But let's say that it's 99.99999% accurate, all right? And there it is online for free, available for the researchers of the world to do research on what we call dry bench research, which is on the computer on these databases, these mega databases. That's the Human Genome Project, the Human Genome Project, right? That happened over the turn of the century and the millennium. Now,
even dangerous.
what is?
You're basically creating another species.
Right, yeah. I mean, images of Frankenstein come to mind.
Yeah, it's like you're going to have a smart,
smart mouse or a
can
smart.
you imagine?
Yeah. Well, this would be.
I mean, if you go
Right, who's going to get it? The good old mouse is going to get it, right? Because that's the lab animal of preference.
The good, I think.
Right.
There's no evil in it.
Exactly. Or the rat, yeah, because the rat's a little bigger and easier to manipulate at the larger level. Right, but the whole point is that the intention of this, right, which is a good intention, is that what is known as the Human Brain Project, I don't know if you heard about it, it's been launched, and it's an international effort to try to learn everything possible about the human brain, beginning with the molecular atomic structure of the synapses and the physiological events of the brain, from all the way down to the molecular, up to the cellular, tissue, organ, and system level, all of it, including psychology, including sociology, everything that can be known about the human brain. That is the Human Brain Project that is going on right now, all right? And it's going out by many different labs throughout the world. It's a interdisciplinary and international project in the full sense of the word. So there are certain ethical things that are going to be kicking in when we talk about the Human Brain Project that should be done or should not be done, especially in view of the Nuremberg trials. What was the Nuremberg trials? The Nuremberg trials was the trial after World War II that tried some of the officers and some of the researchers that were involved in the Holocaust and the whole experimentation of Nazi Germany and Hitler. Here are the judges, and here are some of the accused, and the military police, and those were military trials. But Nuremberg was very famous for that. You can read all about it. They took place precisely in Nuremberg, Germany. Okay. And what was going on here? One of the things that came out of the Nuremberg trials, remember during the Holocaust, it's estimated how many million Jews were killed in the Holocaust? Anybody have an idea? About 6 million. 6 million Jews were killed in the concentration camps, all right? Jews. But it's estimated that about 12 million people were killed in the concentration camps during Nazi Germany, during the Third Reich. And so who are the other 6 million? Because that's the insanity of Hitler that was expanding, right? Beyond the Jews, who were supposed to be the cancer of the earth, and that's why they had to be eliminated, according to his wicked mind, well, anyone who was defective. So after the Jews, he went after, or in addition to the Jews, he went after, who were the other obvious ones? The gypsies, or the homeless of Europe are the gypsies. who are now called Roma because many of them came from Romania. But the gypsies were also contaminating society. They're obviously ignorant and dirty, and the men play poker and drink all day long, and the women and their babies are out stealing in the city and all that. So they had to be eliminated. And then he expanded also the handicapped, the deformed. They were contaminating the earth because they're going to pass on these defective genes. And then also homosexuals had totally disgusting orientation, so they had to be eliminated too. And he went after the homosexuals. And then he started going after religions that were critical of him. So Catholics were also exterminated, including the saint in the concentration camps was the Polish Maximilian Kolbe, all right, who was actually evangelizing inside the camps and everything else. So some saints came out of that. So those are the other 6 million. And he was just expanding. He got so schizophrenic that at one point, the very generals of his Reich, of his rank, were questioning themselves if they were Aryan enough. You know, if they were blonde and blue-eyed and fair skin enough to be pure Aryans. Because it was coming down to anyone who's not a pure Aryan is going to be eliminated sooner or later. Because they were contaminated. Aryan, the Aryan race, right? The Aryan race was the Deutschland über alles, right? Germany above all. And so they were doing genealogies in secret. These are the closest circle of Hitler and everybody else, of course, to see how far back they can trace the Aryan origins and where was the contamination coming in 1, 2, 3, 4 generations back, right? Because it was, and that's why he had to be stopped. He had already taken over Europe, allied with Mussolini in the Axis from Berlin to Rome was the Gothic axis.
And was he in your area?
Exactly. So that's the whole question. All right, that's the whole question. And that's why. So who is? You go back enough, and we all go back to Africa, right?
Exactly.
Mitochondrial leaf, and therefore, so that was the insanity. That was the insanity that was building on itself, and he had already spilled into Africa. That's why this fellow had to go after him, Rommel, and who was it that made the movie then, the famous movie? Went after him in Africa, and he was expanding over here. The U-2, the boats, the U-boats, which were the German submarines, there was already evidence that they were patrolling, they were looking at the coast of the Caribbean islands to see where they were going to land and make a beachhead so that they could invade the United States. There's evidence that there were U-boats, that there were German submarines in Hudson Bay in Manhattan at night with the periscopes looking at the city of Manhattan. So they were planning on invading the United States. There's evidence here in Miami and South Florida where we had blimps that would patrol the coast at night. They're very silent in the dark to see if they would spot any U-boats. That was in the 40s here in Miami. We had blimp, even here, Opelok Airport had a blimp station for these Zeppelins, these blimps that were patrolling the coast in the dark at night for U-boats. So this was absolute insanity. That's why he had to stop, and that's why it took a Second World War to stop him. Mm-hmm. So the trials, back to the trials, where obviously all these people were found guilty of heinous crimes and atrocities against humanity and so forth. The scientific community at that point decided not to use the results of the experiments that were done in the concentration camps. Because the experiments that were done in the concentration camps had the highest rigor of scientific protocol possible. They were extremely rigorous, and the scientific quality of those experiments was the highest standard possible at the time. All right? There was one glitch. There was evidently no informed consent whatsoever. The humans were manipulated as if they were less, because we wouldn't do to cattle, we wouldn't do to animals what was done to these people. All right? With electrocution, with all kinds of manipulation and mutilation. Hypothermia, they would put them in cold with ice to see how long the body would last before freezing. The mother-daughter experiments that were done on psychological to see who would shock the other one with electricity first, and to the point that most daughters ended up killing their mothers with shock, and then they became insane. The daughters became insane. So the horror was just appalling. And so, but done with the highest standard of scientific rigor. And so the quality of those results was unquestionable for science. However, the way that it was obtained, the means, was horrific. And so the scientific community at the time decided not to use those results, right? Which could have advanced science much more at the time and so forth, all right? So those were the Nuremberg trials. Why?
Like they did found, like they burned the books or?
No, they were,
The results.
I don't know exactly, but yes, I don't even know who owned them and so forth. You get into the legal aspect of who owned those results and so forth. But yeah, they could have been burned. Then you get into the human factor of some of those may be stored somewhere and they have historical value, right? For once, I just kind of thinking out loud here, you know, that the Jews, they live insofar, one aspect of Jewish theology is that their eternal life is in the memories of the people, of the living. In other words, the dead continue to live in the memory of the living, all right? And therefore, it is essential, it's imperative for the Jews, the living Jews, not to forget the dead Jews, and especially if in the Holocaust, the Shoah. And that's why they make so much emphasis on it. That's why there are monuments, that's why there are museums. There's a Holocaust museum here in Miami Beach. Have not been to it, but I've heard it's very impressive. I've been to the one in Washington, D.C. Very impressive. They even have there, well, some of the speeches going on of Hitler in black and white and a little video thing. They also have one of the carts of the many hundreds and hundreds of cattle carts, the train carts that were used to push people in there like this. Many of them didn't survive. They were asphyxiated to death on the trip and so forth. So very, very impressive. The whole purpose of the museums and the memorials and all this and the names and walls is so that we don't forget, humanity doesn't forget what happened. And the whole thing, never again, right? Never again. We should not go there again. So anyway, I'm saying this because of the human genome, the human brain trial, the human brain project. When we talk about experimenting with the human brain, well, naturally, a human brain is inside a human being. And so we need certain parameters, right? Because now at this point, these are born human brains, these are born human beings, and therefore, all of the protections of law should kick in, beginning with informed consent and so forth. So there are obviously certain things, many things that we cannot do with a live human brain, because that live human brain happens to reside in a human being who is now protected by law. And so if I am able to generate a human brain in a mouse or a rat or whatever, that rat I don't need to get informed consent from that rat. Okay, and now I can do some experiment on human brains in an animal. So that was the thrust of this. That was the desire. And I can tell you quite candidly, the date is extremely significant because it happened that this human brain project started under the Obama administration simply because of the 2 terms and the 8 years and so forth, and it kind of coincided. And he was in favor of it, and the American researchers are in collaboration with the whole world, with other labs throughout the world, where this project is moving forward, okay? With ethical guidelines and the IRBs, you know that whenever there is a human subject of experimentation, there has to be an institutional review board. Even we have it here, Little St. Thomas. When we get, the typical thing is we get all our EDDs. What are the EDDs? The educational doctors. Well, to do a doctorate, one is supposed to do original research, the dissertation. And typically with education, it involves children, minors, so they need a review board. And we're constantly getting proposals of doctoral dissertations when we review, and we have an institutional review board, an IRB here at St. Thomas University as part of the accreditation system and so forth to review all of these proposals for dissertations of testing whether a child, if he has breakfast or not, doesn't make a difference, if he pays attention or falls asleep in the class, whatever, to get some research done so they can do their dissertations for their doctorate, right? I'm just giving you one example. So there are IRBs in hospitals, in universities, in many places, they review for human subject research. And the basic thing is precisely that informed consent. In other words, not just consent, but these students have to tell the parents what is going to happen to the child when they take the test or do a little survey, whatever it's going to be. So it's very rigorous, as it should be, because we're dealing with human subjects of research. Okay, so it would be very convenient to have an animal with a substantially human brain. Doesn't have to be a full human brain because a full human brain doesn't fit in many other mammals, okay? But it has to be just functionally, substantially human, where some human processes can be tested. And once you know it, then once that goes through the relaxation of that ban, then they can start tweaking it and they look at sensory areas, they can look at the speech area, they can look at the visual area, the different parts of the brain. They could eventually construct just that part of the brain, of the human brain, in the mouse or in the model that they're going to use. You see? Start tweaking it and tweaking it. Why? Because it can be done. It can be done, and with CRISPR, even more so. CRISPR, which is the insertion of specific genes from one species into another species in a particular region to express. So I can get, let's say, hypothetically, identify the human genes that code for speech understanding. All right? They're also going to be in the brain and the cortex of the brain. There have to be specific genes that code for specific proteins that do that function, of the 20,000 genes that we have approximately. I take those genes and insert them into the mouse brain in the same region of speech interpretation of the mouse, which is going to be little songs by the way they sing. The mice, it was discovered a few years ago that at night, in the lab mice that are being raised in these holding areas, labs, at night, the male mice are singing for mating songs, mating rituals. Typically, songs are involved in mating to the females, who can be in other cages and so forth. And there's this whole symphony going on of singing at night in these clusters. But no one had ever heard of it. And at some point, I think it was a student or something, put, just for the kick of it, put to detect sound waves that are beyond the hearing range of the human, like the dog whistle, the dog whistle that has a high pitch. And he got a whole symphony of noise that was occurring overnight, and finally figured out that the male mice are singing to the female mice all night long.
Serenade.
Yeah, it was a serenade. Exactly. I know it's an interesting discovery, but no one could hear it because it's a pitch that is outside our range. Just like infrared or ultraviolet that we don't see in the visible spectrum, it's outside of the visible spectrum of light, right? Of visual for the human eye. Anyway, back to this. Here's the proposal. So this is proposed scope of Chimera. This is to establish the steering committee for the relaxation of the ban, all right? Research in which human cells are introduced into post-gastrulation non-human mammals, okay? So non-human mammals, excluding rodents, so this would not be done on the rodents, where there could be substantial contribution to animal brain or substantial functional modification to animal brain. That's the way it's worded. When we translate this into common English, is to create a substantially human brain in a non-human. That, by the way, is not going to be a rodent. So the next animal of choice is going to be probably a primate.
A rabbit, yeah.
Could be a rabbit or no? It's a rabbit or rodent?
It's a rodent.
Exactly. The monkey. Maybe the rhesus or maybe the chimp, I don't know. But it's most likely the primate, which is closest to us genetically, right? With that 99%
But the
compatibility.
purpose what is followed to have human brains
Yeah,
for research without consent.
exactly. On an animal, where you're not going to obtain consent from an animal because the animals, first of all, it's impossible for the animal to give consent. But then, so what are the protocols for animals? This begs the question of the protocol for animals. The protocol for animals basically is that they're treated humanely.
Yeah.
You know, and when they have to be euthanized, we call it sacrificing the animal, euthanizing the animal. Well, before euthanizing the animal, we anesthetize the animal so the animal doesn't feel pain or suffer and so forth, but we end up killing the animal for the research, because these are the animal trials that are done typically in medication, in drug trials, before we get to the human trials, because it's ethically important to test on the animal before we test the drug on the human and make sure that it works on the animal before, you know, we expose the human to risk.
When I was in medical school,
Mhm.
I used rabbits and dogs.
Yeah, dogs are like it.
For surgery.
Mm.
Oh my Jesus.
Yeah,
I mean,
Right.
they were you put them to sleep, and then they had to be.
euthanized humanely.
Yeah.
Were they just asleep or were they actually euthanized?
No, they were just asleep.
Only asleep?
I mean, they were sedated. Like
Sedated,
surgery.
exactly.
Yeah,
They were anesthetized.
they were anesthetized.
Right, and then you work on the organs and so forth to practice. Sure, because you're not going to practice on humans, right?
No,
They-
no,
they
and
serve-
there are limits too.
Yeah,
There are
right.
limits. I remember when I was doing my bachelor's, I was working with mice with malaria,
Hmm.
and there was this girl, she was doing a PhD, and she was doing something of a protein. But
Uh-huh.
the thing is that she was using rats because she needed a large amount of blood.
Right, yeah.
But the thing is that it was like 6 years, she was still doing the project, and
Wow.
she didn't get to anything, and she was just killing rats, and the board said, you have to stop.
Right.
You're using like 15 rats weekly, you have to stop, and they just take out her project because they say
Yeah,
that's not ethical. I mean,
exactly.
you're not
It's
getting
inhumane.
anywhere. Your project said that you were going to use only 40 rats
Total,
in
yeah.
3 years,
Mhm.
and you're using too much, so it's not ethical for the experimental animal, and they shut down the project.
Exactly. You see, it's
There
inhumane.
has to be a respect for the animals.
Exactly. And so that's what we call the humane treatment of animals, right? And there are some organizations in society that would like to ban animal experimentation altogether, like PETA and these other groups, right? In my opinion, it's excessive, because then what are we left with?
The humans.
Exactly.
And
Exactly. We're gonna-
they will approve research, but not with animals.
Exactly.
Pretty sure we're gonna get there.
Right. Well, typically what will happen is the embryo will get it, right? The human embryo, because the human embryo cannot complain and it has no protection under the law, or very little protection. So typically the human embryo is going to get it if we don't experiment on animals. But the animal experimentation, you're right, and it has to be justified and has to be done in a humane way, and the protocol has to make sense and so forth. Mm-hmm. And I was telling the undergraduate class, because they also, certainly they would freak out about things like this. I say, well, before we experiment on humans, we're going to experiment on mammals, on, let's say, dogs or mice, whatever. Before we experiment on mammals, we're going to experiment on flies, on fruit flies, for example. Okay, well, that tones down a little bit on the ethics, because we can certainly use many more hundreds or thousands of flies before we go into any mammal. And before flies, if we can do the experiment on bacteria, we're going to use bacteria first.
Yeah.
And work it out on the bacteria to make sure that in the bacteria it works. Then we'll move to a fly model. And once it works in the flies, then we'll move to a mammalian model. And finally, at some point, if it's a drug development, after several years, we'll move into a human trial. But you better believe it that we're going to have informed consent from the humans, okay? Because they're not guinea pigs. And so that's how it's worked out. And the benefit of that is, for example, just antibiotics that are used throughout the world and so forth for healing. Mm-hmm. But
and a lot of the, for example, the medication trials
right.
are done with humans.
Right.
You know, there are persons that go there, you know, they sign a consent.
Exactly.
Whether participate in the study, and some of them know, then nobody knows who's going to be taking, you know, medication. They usually go
And they pray
right.
to be the placebo.
Or the placebo, right?
I know,
Exactly.
but, that's the way they find
That
out.
has to be blind. That's what they call double blind, and the individuals cannot know because they want to see the reactions, and if the reactions are actually physiological or psychosomatic and so forth. So those are, yeah, that's the rigor of the protocol. Okay, and that's what goes through the review.
Yeah, for me it was very difficult when I started working with animals.
Mhm.
Because, you know, this little cute mouse, and I was like, I'm
Right.
hearing, I mean, they're creatures from God too.
Exactly.
So the manager of the Utilitarian, he sat with me and explained to me,
Yes.
I mean, we have to do this.
Right.
I mean, what's the end? Through disease, you see all
Exactly.
these kids in Africa who are suffering from malaria, so we have to test them out. As long as you do it with respect,
That's the
you
whole
use
thing.
the protocol, you don't abuse, you're fine. And of course, if you see you're getting, you cannot be obsessed in something and just kill a mouse and it's not going no. You have
That's
to know
exactly
where
right. Yes.
And there, it's a point where it's okay.
Exactly. In other words, exactly. You try to use the minimal amount of animals possible, right? These animals, after all, are being raised in the lab anyway, so they wouldn't be, we're not pulling them out of nature. For the dogs, typically stray dogs that would end up being euthanized anyways, they're going to be euthanized and incinerated. And so it truly is, it's practical, but it's not utilitarian. It's not just doing experiments, excessive experiments on animals, or using an excessive amount. It has to be justified. And if it's not working, then it's time to review the protocol, and there's something wrong with the protocol. There's not something wrong with the mouse or the animal. Okay. So this, I can tell you, it is super regulated today to the point that if those protocols are not followed, the whole lab will be shut down, and sometimes the whole building will be shut down, right, by the regulatory agencies. Even to the point of, I mean, the protocol for raising mice, for example, in the lab, it's huge. It's a thick book. It has to be followed rigorously. I know, to give you an example, when the mom drops the litter, all right, she'll have, I think the cycle is every 3 weeks or so. They can raise a new litter, so it's very fast turnaround, and they'll drop 4, 6, 8, 10 mice. The father has to be taken out because the father will start eating the young. All right, and that's inhumane. So when does she drop the litter? It could be 3 o'clock in the morning. Someone has to be there when they expect her to parir, to give birth. Someone has to be there to take out the male, separate them, because otherwise.
Yeah, they start eating.
That's right. And the following morning, that lab will be shut down, and that whole building will be shut down because they didn't follow the protocol. It was inhumane. See? And that's how rigorous it is. And it's a good thing. But so this was the thing, and let me show you, okay. Right, so this was done in August of 2016, what happened in November of 2016? So the 8-year term of that Democratic president was over, and there was a toss-up between Mrs. Clinton or Mr. Trump, right? And it could go either way. So why time it here? Just a couple of months before the possibility of an administrative turnaround in the opposite party, which would certainly ban this, because this would be not according to the party platform of the Republican Party. And so just in case they wanted to do this, NIH wanted to do this before a possible turnover in the next party line at the federal level. So the timing was.
We didn't know those things.
Mm-hmm. Exactly. So this was put on but because this is a public trust, or this is a public run agency with our taxes, they have to, by law, publish this stuff. So they published it kind of under the radar. Someone picked it up and was being sent around to some bioethicists. Someone sent it to me. And I can tell you, I spent days sending out emails from an extensive email list that I have in little clusters and they gave a site when one could click and give the opinion, and they were hoping to not get many opinions from the general public, because if this really got out into the general public, the natural outrage of the disgust of trying to generate a substantially human brain in an animal that is what we call the yucky factor. Many people intuitively, exactly, have a conscience to say we shouldn't go there. So they would have been. Exactly. That's the gut. That's really the gut conscience saying we really shouldn't go there, folks. I may not be able to articulate it scientifically or theologically, but we really shouldn't go there. That is way beyond what is reasonable and decent.
And what is the status for now?
So the end result is that NIH, which gave a very little window for response and comment and so forth to the general public, got such an overwhelming no to this that they rescinded the the relaxation of the ban. So the ban still holds for not doing this. In other words, they were not able to relax the ban, which means that they could not release the federal funds for it. But this says nothing about state funds, and this says nothing about private funds. And so the labs who were insistent on doing this, they had to shift to state or private funds.
So if there's a millionaire that says, oh yeah, they can do it?
Yeah.
That's right. And then what happens again, the economy is driving it because the biotechs that get into this, they are high risk in the market, but if they hit it, then there's high return. And so some high risk investors look to put their money there because they expect to lose the money, but if they don't lose the money, they're gonna cash in big time. 100 to 1 or whatever is gonna be the investment, right?
And where can you see this kind of things, Father? Because I didn't hear that in the news.
Exactly.
Is there a website?
You have to search, you get in touch with the National Catholic Bioethics Center, you have to have your radars out for this kind of stuff. You have to, be part of a network where we can network each other. And for sure, if ever I spot something like this, or if you spot it, well, now we have each other's emails, right?
Because I never heard of it.
Well, maybe I do hear. I don't remember what it was, but I do remember my husband being very annoyed and posting a million things on Facebook. Like, everybody's so concerned about elections, and look what they're trying to slide by us. And
Right.
It could have been this. I don't remember exactly if it was something else, but like a lot of things go under the radar.
They do. They do. Now, this is, of course, this is after the fact, but you can go just to, whenever I have a doubt, I punch it in, right?
Yeah.
Into Google and just put, you can put human, animal, chimeras. Yes, it's supposed to be because these are federal agencies. Okay, so human animal chimeras are getting to US research farms. This is from MIT. This is from the BBC, the birth of half human, half animal chimeras. Okay, so the pig hybrid, this is what I'm going to talk about now to show you where it actually happened. And they did the experiment. They didn't use federal funds because they did the experiment here in the United States at Max Planck, which is the institute in California. And then they sent the embryo to be implanted in Spain. And they used Spanish pigs because of jamoncito in Spain and lechoncito and bejugo. They have a lot of pig farms in Spain and they're very successful at breeding pigs. So that's where the embryos were sent. But the embryo was manufactured. The chimera was manufactured in the US because that in itself was not illegal. It was the implantation part, okay? Just to prove they get around. They get around. If there's a will, there's a way. And let me show you, that's the next slide here. So here's the actual paper on Cell, which is one of the premier journals, scientific journals of the world. And here's the experiment. We have a human. We generate a blastocyst by IPS or an ICM, remember, induced pluripotent cells, right? Yamanaka. In other words, from human cells, neuronal cells, because these have to be fully differentiated, neuronal cells, were de-differentiated to the blastocyst stage, to the pluripotent stage. Those cells, which you see here now, are inserted into a pig blastocyst. All right, let's see if I can make this a little bigger. This is the mechanics of it. Human neurons are dedifferentiated or undifferentiated back to blastocysts through IPS, induced pluripotent cells, right? And those now pluripotent blastocysts, which came from neurons, so they're neuronal line, right? When they redifferentiate, naturally they're going to redifferentiate into neuronal lines, human neuronal lines. Those cells were inserted into the ICM, the inner cell mass of a pig blastocyst. So that's the chimera here. This is the chimerism. We have a pig blastocyst, we have a pig embryo that has been inserted with neuronal cells or neuronal precursors that are also pluripotent. And now that chimera is inserted into it's called a sow, I think, a female pig, right? To gestate to a certain stage. Of course, this is just what we call proof of principle. They just want to prove scientifically that it can be done. They're going to destroy, they're going to kill this embryo, they're not going to take this embryo to term.
What's the name of it for?
Proof of principle. Principle. In other words, they are proving scientifically that this works, that they can take human differentiated cells, in this case, the most differentiated cells of the body, which are the neuronal cells, right? And undifferentiate them, dedifferentiate them back to the pluripotent stage by using IPS, and then insert those cells, those neuronal precursors, into an animal embryo, an animal blastocyst, in the ICM, which is what becomes the embryo proper, but it's patchy. You see, it's still kind of shotgun because those human cells embedded into the ICM in different regions of the ICM, but it's still shotgun because at that point it wasn't known, it was not known where those neuronal precursors were going to express.
Right.
Could be anywhere, and you can see here diagrammatically they're shown to express in the shoulder, in the what is that called, the hank, the hip area. Apparently they express also in the heart area of the little embryo. But here's the chimeric embryo, which of course is mostly pig, but there's some tissue in this pig that is human.
What's the point of that? Like, what are they going to do with that?
Exactly, it's still shotgun. Eventually, the idea was to identify what region of the ICM becomes the brain, which is not all that hard because take it to the next stage. After the blastula, what do we have? We have the gastrula, which is the 3 germ layers, right? And we know that there is, through invagination, there's this neuralization that happens. The neural cord is going to come from the inpouring of the ectoderm. So if we get to implant the human cells on the ectoderm of the gastrula, some of that ectoderm should pour in to form the neural tube, and the front end of it is going to become the brain. That's where cephalization occurs. So there's a region. So if this could be done instead of the blastocyst, if this could be done in the gastrula, we can target at least one of the 3 germ layers. And the one that would be of choice for cephalization would be the ectoderm.
Yeah, and I suppose they say, no, we're not using human ICM.
Exactly.
We're not doing Vitro in humans, so it's
Exactly. It's not a human being. Exactly. It's not a human being at all. This is just human tissue that is being transplanted.
I mean, they're not using ICM. They're using the IPS.
Yes, of the human.
Of the humans.
The ICM is a pig. So that's why it's a substantially human brain in a pig, in a non-human, all right? So this is just to prove it's crude, really, when you start looking at the details of it, because it's shotgun, it's not really necessarily in the brain. But again, how many pig blastocysts can we get? Any number of thousands of them, or hundreds of thousands. I mean, I'm sure there are at least hundreds, if not thousands, of pig farms in Spain, and that's just one country, and this is all in vitro anyway. So again, it's the manipulation, the shotgun. This all starts with a pig egg, right? Fertilized by a pig sperm and so forth, so you can get any number of blastocysts. For the potential of knowing what happens in the human brain at the biochemical level, imagine the millions of dollars that can be poured into this. So once it gets established, the protocol is fairly easy to repeat, to imitate in other labs of the world. And at some point, just by shotgun, they're going to get some of those human lines into the brain of the pig, just on average, okay? Statistically.
But if you develop, he can have like a human brain product?
A substantially human brain. A fully human brain doesn't fit in a pig, right?
Exactly.
Doesn't fit. But we're not interested in the full human brain. In fact, the white matter, who cares? It's mostly the gray matter. It's the surface, it's the cortex, where thought process happens. And even that, you've seen the maps of the human brain, right? It's mapped out, the whole cortex is mapped out. That is well known already and established. So there are centers of writing, there are centers of motions.
Physiological, no?
Yeah, physiological and biochemical, as much detail as they can get. You've seen these images where they have where is it?
So they're not going to have like a super smart pig, no?
No, not at all, not at all. If anything, it's going to be a schizophrenic pig. Okay, what is the pig going to think? It's just part of the brain. So who knows? I mean, will the pig eventually understand human speech? That's a long
that's
shot.
What I'm saying, or maybe he start talking. You don't know. We don't know.
Yeah, but they'll also need the accessory organs to do that, right? They will need the vocal cords and the vocalization and all that. So we have to think always functionally. Let's see here. This
higher up, maybe
higher.
on the right.
But wait, I just saw something. Human brain.
That one on the right.
Human brain mapping.
That one.
Could be. Well, this one, yeah, this one has the different lobes.
Oh,
But
higher up you had
it could be. I was just looking. I've seen those images where they have on one side of the brain they have the region, on the other side they have what is responsible for. Let me put here brain. Brain
How about
and
how human brain mapping can help you?
could be, yeah. That's it. Well, I don't know how to retrieve what I'm looking for. No. It's not showing. Sorry. I'll try to find it during the break and then try to show it to you. Okay. Let me go back. But here's the actual experiment. Start with here's the blastocyst. Here's the target area, which has already been, the zona and the corona have been compromised so as to insert. And here is the little cannula, which is just a glass rod, very thick, to the thickness of a single cell. And here are the neuronal precursors. All right, so that's going to be inserted here and deposited inside. Here's the ICM. And here's the vacuum suction holding the whole structure in place. Okay, so that's the actual micrograph. Oops. And down here is the reference to the article. Again, in Cell, you can follow it. So it's happened, and that's the whole point. If it can be done, it will be done sooner or later. The technological imperative coming out of the utilitarian camp. Moving forward. So in a sense, we can say that this is growing a human organ in a non-human, right? In an animal model. Happens to be the brain, or in the other case, it would be the ovaries. But how about some positive aspects? And I'm going to try to shift here a little bit into the positive. How about growing other human organs that are not as controversial as a brain or a gonad in animals? This was in the 1990s, the vacante ear, all right? Someone was talking about nude mice last class. Well, this is an example of a nude mouse, hairless. They're just cleaner, for example. They also happen to be albino. This one has a little red eye, so this one happens to be albino. But they're hairless and they're easier to maintain clean and so forth because hair is very dirty. And since they're looking at a structure growing underneath the skin, so the idea was to have the mouse grow skin over a structure. This is not an actual ear, this is the ear lobe. As you can see, there's no connection inside. It doesn't connect to a middle ear to an inner ear, right? This is just the outer lobe. And essentially was to prove that one could insert a scaffold underneath the skin of a mouse and have that scaffold grow on top for regeneration of the lobe itself, right? But not the full ear. So it's not that this mouse is going to hear through this human ear, human words or anything like that. But it's a scaffold, which scaffold today can also be made in 3D printing with biodegradable substances that after a while, once the human skin has grown over it, that biodegradable substance will be digested away, and you have the reconstruction, essentially, of an ear lobe, all right? Which could be a good thing because you can expand that then to the nose and to other parts of the body and eventually get more sophisticated with potentially actually growing organs, human organs, in a non-human. As, for example, a human pancreas in an animal model could potentially have an unlimited supply of human pancreas to be transplanted without having to wait for a cadaver, a human cadaver, to do the transplant with all the complications of transplantation.
Like we used the porcine valves for valves.
Yes, porcine, exactly. Heart valves for years now. It turns out that the pig, going back to the pig for a moment, the pig's heart is very similar to the human heart in size and function and so forth. And the valves happen to be pretty similar in size. And so for many years now, for decades, pig heart valves have been used. Of course, they're clean, they're washed and stripped of most of the tissue and then transplanted to replace defective heart valves, human heart valves, right? Either the bicuspid or the tricuspid. So that's a positive thing. And the pig is going to end up in lechon asado anyway, so might as well profit from the heart, you see? So that's, this is on the positive side. And we're not opposed, in other words, to just any organs being grown on animals, human organs. There could be something good because I think it's pretty intuitive that the human soul does not reside in an earlobe or even in a pancreas or any other, or even literally in the heart, the human heart, because organically it's a muscle, right? Okay, what do we have? We have prenatal diagnosis and fetal surgery to go. So perhaps we can break here. Yes. Why don't we break here for a moment? Let me just make sure. I think we only have 2 more topics. Yeah. Okay, so we got prenatal diagnosis and fetal surgery. So let's break here. And we'll come back in a few minutes, okay?
I'm sorry that I asked so many questions.
No, it's good. It's exciting. Yeah. They are, and that's the whole point about bioethics, you know. Look, I always have this principle. The only wrong question in my book is the one that's not asked. Because at times it's awkward to formulate the bioethical questions, but unless we get it out in dialogue, we really won't be able to get at an answer. So feel free anytime. Okay, let me pause this. Okay, folks, welcome back. Let's look at 2 more topics here. First on prenatal diagnosis, and the other one on fetal medicine and surgery. So prenatal diagnosis has been going on for quite a while already. PND, or just PD. It actually probably goes back to the 90s or even earlier with sonogram. So we have some techniques that are non-invasive, others are invasive. We'll look at those. So we'll look always first at the clinical part and then the bioethical evaluation. So on the clinical side, some are non-invasive or minimally invasive, and then others are more invasive. The easiest one to do is the ultrasound, the famous sonogram, which is now done standardly on pregnant moms, because the first thing that the doctor has to determine is the gestation age, the time of pregnancy, right? As that pregnancy develops, to estimate if the pregnancy is going well statistically compared to other pregnancies with body weight and so forth. And then ultimately the time of arrival, the time of birth, to calculate that. So ultrasound now reveals also other things about the human embryo. It reveals whether the embryo is deformed or not in the proper position, and as the pregnancy develops, whether the fetus has the organs properly developing, including the brain, the appendages, the legs and arms and so forth. They can get down to details of the sex, right, of the embryo. So all that information is kind of a two-edged sword, because what is the information going to be used for, right? Other minimally invasive, certainly non-invasive for the embryo of the fetus, but minimally for the mom, is the maternal blood serum, because some proteins go from the embryo to the mom or the fetus through the placenta, and then those proteins can be analyzed, like alpha-fetoprotein, which is one of the growth proteins, and the levels that are normal or abnormal in the mom's circulation. It's known now that quite a few substances move from the fetus to the mother and vice versa through the placenta, including immune cells, actual cells from the fetus that end up integrated into the mom's body, a few, but some real, right? So it's a little bit kind of a chimerism that occurs there between the embryo and the mom. So those things can be detected in the bloodstream and monitored for proper development or not. A little more invasive are the amniocentesis and the chorionic villi sampling, or CVS. Amnio is to take out some fluid through transabdominally. A needle is put through the abdomen of the mother into the amniotic sac, and some of that amnion, some of that fluid is extracted into a syringe, small amount, and in that fluid will be cells from the fetus that are sloughed off, mostly skin cells that come off from the skin of the fetus into the amniotic fluid. We're constantly shedding skin cells, right? Those cells can be sent to the lab for karyotyping, for chromosomal analysis, for genetic analysis, to see if there are any abnormalities there at the genetic level. And there's an increasing number of genetic abnormalities can be tested in the lab, all right, with human tissue. So that's amniocentesis. CVS, chorionic villi sampling, is a little more invasive. These have some risk of spontaneous abortion, of triggering a miscarriage. I think for amnio is about 5%. For chorionic villi sampling is about 10%. At least a few years back when I was doing this research, it was double the risk of inciting a miscarriage, because CVS involves taking a little bit of sample of the chorionic villi. Let's look at chorionic villi for a moment. The chorion is a layer of cells outside the amnion that is actually embedded into the part of the placenta, and the villi are these little hair-like structures that are part of the placenta near the surface. And so it's a little more invasive. It actually involves taking out some tissue. Let's see, a little bigger diagram. Well, maybe the first one is got more than what I want to show, but here's the placenta, and it's taking out some of the villi, some of the hair-like cells of the placenta. Let's see what's a little closer. Let me just enlarge this for a moment. You see? And then those, that little bit of tissue is sent to the lab. That is fetal tissue because it's part of this side of the placenta. Okay? And that is also sent for sampling in the lab. But by sucking out some of that little amount of tissue, as small as it is disturbing the placenta, right, and compromising the integrity. The plug, for example, is compromised and so forth. So it could trigger a miscarriage. And so it has to be motivated. It has to have a significant reason for doing it. And what's the significant reason on the positive side? That's the challenge. I only bring it up because this was one of the paragraphs that was a positive paragraph, if you will, conditional positive, in the Ethical and Religious Directives, the ERDs. It's directive number 50, actually. So, and because it had some angle of positive, I ended up doing my dissertation in Rome on this document that had just come out in the 90s when I was in Rome. And so prenatal diagnosis, which was new at the time, is permitted when the procedure does not threaten the life or physical integrity of the unborn child or the mother, and does not subject them to disproportionate risk. Also, when the information that is provided is not used to procure an abortion, right, but rather to do some kind of preventative work or care on the fetus if possible, or at least to alert the parents that they're gestating a child with some kind of defect, all right, to get ready to prepare. And also, obviously, informed consent, which in this case had to come from the mother for sure. So it is definitely not permitted, just to be clear, when that information is going to use for the intention of aborting a child that has some kind of defect, right? Because that would be eugenics, that is killing someone for a deformity. So that's a little paragraph there, is paragraph number 50, or directive number 50 of the Ethical and Religious Directives. And so we're very clear, at least from the Catholic perspective, that this prenatal diagnosis should only be done when it benefits either the mother or the child or both, but not to do any kind of harm. And so one can actually question if an invasive procedure would be justified, to what extent, since being that an invasive procedure, by definition, it has some level of risk of triggering a spontaneous abortion or a miscarriage, which would be lethal for the embryo, right, for the fetus, amnio or CVS. To what end, in other words, we'd ask. What do we do with the information if the child comes with an anomaly? It's not proper to kill the child, so why find out? Just so that the couple gets ready? Well, actually, these two techniques, in fact, all these techniques are being used today in a secular environment as a search and destroy mission, as it were. Typically, the one that gets selected out are Down syndrome children, to the point that some Scandinavian countries have declared as a national goal to eliminate the Down syndrome population with the unborn, because they're not going to touch the born yet, because they supposedly have some legal rights, right? They're born, and so they're recognized as human persons. But they're going after the unborn, and the idea is to eliminate Down syndrome in their country. However, when you think about it, it's totally very short-sighted, because it's not that Down syndrome is necessarily inheritable. Typically, they're sterile, right? They're infertile, so it's not like that. It's not generated that way. What is the typical case of Down syndrome, or most of these chromosomal abnormalities? Aneuploidy, in other words, old eggs, just simply old eggs, that women are waiting to the 30s or the 40s to have the children. And so even if they were to eliminate all of the Down syndrome embryos in today's population and generation, the next generation, if the women have old eggs, they're going to get Down syndrome again. And so they have to continue to search, target, and destroy these Down syndrome children in utero, okay? So then the next step is to generate a social environment whereby any mom who is giving birth to a Down syndrome child will be socially accountable for raising that child, and she exposes herself to lawsuits of wrongful birth or wrongful life.
Oh my God.
Yes. So the child can sue the mother for having given him or her birth onto a Down syndrome life, all right? And if the child cannot sue, then an advocacy group can sue on his behalf. Yes, this is in Scandinavia. I think it's Sweden, but I'm not sure the country. We've seen some of those suits here in the United States also. They're called wrongful birth or wrongful life. So far, they haven't got much traction in the judiciary system, so they're dismissed as frivolous, right? But the minute one of those suits gets traction and is awarded, then that sets precedent for the kind of law that we have, which is what is called tort law, whatever it's called. I'm not familiar with that, but if one case has been settled already, then it can be used for the next one and the next one, and then they build a culture whereby, and even just socially, the pressure on women who have children, who bear children who are abnormal, the social pressure is, how dare you do something like that? It's injustice, you're abusing this child, and so forth. Okay, so that's the ethos. That's the ethos, which is the ethical environment of today's society in a secular way. P&D can feed into that, unfortunately. So it's a two-edged sword, right? All right. However, on the positive side, how about some fetal medicine or fetal surgery? All right. Well, we have it. It's happening. There are institutes and foundations and organizations that are already involved in the practice of fetal medicine. There's even a journal now. Okay? And it's happening because we can do it. And so, in principle, we should do it if it's something good and positive. All right? So this is just, for example, this is just March of last year, so it's less than a year ago, a workshop on maternal fetal medicine. It's happening, it's out there. There's a whole journal on it. And so a few examples of the stuff that can be done. Typically, there are little shunts and bypasses and other stuff, or some kind of surgery can also be done. For example, spina bifida or meningoceles on the spinal cord. On twins, ablation of the common placenta so that one twin does not, when you have discordant twins, that one twin is absorbing, has the bulk of the placenta, and the other twin is miniaturized because it's not getting enough nutrition, right? That pathway can be severed to favor the smaller twin. This is an actual surgery occurring. Here is the uterus being opened, and here is the unborn. These are actual photographs. This is a little shunt they put into the lungs for proper development to have the proper pressure grow there. They've done little shunts on the heart. They've done shunts on the brain for hydrocephaly and so forth. This is a very famous photograph of one of the earliest fetal surgeries done. This is the uterus here, and the womb was actually, the pregnant womb was actually disconnected from the mother and put on the operation table to operate on the very sterile conditions, of course, and with micro manipulators or micro tools. And once the surgery was done, the womb was sewn up and reattached to the mother. Amazing. And this picture started circulating around the world because as the surgeon was getting ready to suture up after the surgery, the little fetus pulled out the hand and grabbed to the first thing that was there, which happened to be the finger of the surgeon. And it's, I mean, so now it has become kind of popular, and whenever you see many of these versions of this photograph, okay? And now surgeons are just putting their finger there until the fetus grabs, but the first one was kind of spontaneous. The fetus actually reached out and just grabbed. So we'll end it on a positive note like this, that it can be done and for the good. So it's out there.
That photo for pro-life.
Yeah, they do. I've seen it.
Yeah,
Yeah. Not that one.
Yes.
Similar.
Something like it,
Yeah,
right?
It's alive, it's a baby.
Exactly, right. And so what I did is I capitalized on this too in my dissertation, and I actually, the proposal of my first doctorate in Rome on moral theology was the fetus as a medical patient. If the fetus is a medical patient, then that patient has certain rights, just like any other right? The HIPAA and everything else should be kicking in. Because now, how can you have a patient that is a thing or a product?
Right.
Yeah. Medical patient, by definition, they're individual human beings, all right, subject of human rights. And so just like we need certain ethical protocols to experiment on born patients, this angle of it, just on the health of it and the fact that we can do medicine, we can practice medicine on fetus, that makes the fetus a medical patient. And from some perspectives, it's an ideal medical patient. Think about it. Fetus in the womb, right, has a sterile environment, is being nourished by the mom, the placenta. So the mom is a hospital. And the tissue is pristine, a young tissue that will regenerate and grow well, all right, because it's just barely forming. So for many angles on the clinical side of it, the fetus is kind of an ideal patient. They don't spend extra time in the hospital. They're not exposed to the superbugs of a hospital stay, how they try to kick you out before overnight and so forth. The mother takes care of the patient. So from that angle, there are many positive things, and this kind of good medicine should certainly be encouraged, and we see it happening in Catholic hospitals and so forth. But obviously, it requires sophisticated technology, and we don't know yet to what extent the insurance is going to cover all this, but to the extent that insurance does cover this kind of medical practice, then that's all positive. There can even be some medicine without surgery on the fetus, certain medications that will go transplacentally and affect the fetus in development. So those things are good, but it's mostly experimental at this point, right? So these techniques, still at this level, is experimental. Eventually, some standard protocols, some protocols will come out and will be standardized with the volume as soon as they can be validated statistically and so forth. But we expect positive things to come out of this. And I think, to me, one of the greatest values is that it highlights the humanity of the human embryo or the human fetus, right?
And when then we're trying to save them from whatever illness or ailment they have, and on the other hand, they're trying to kill them.
Exactly. Yes, exactly. So you see the subjectivism there. As long as the mom wants this pregnancy, then we can intervene and so forth. If the same mom doesn't want this pregnancy, or after the surgery, she can even decide not to, oh no, I don't want him after all. She can still decide not to want him. Okay? And so that's really, in my mind, that's what I call social schizophrenia.
Yeah, or if the surgery doesn't work.
Exactly, that's it. Yes, you're right. If it doesn't work, you can discard him anyway, so might as well go for it, right? Yeah. These are the challenges, and I think that these challenges are going to be with us for some time, simply because it is a pluralistic society. We're supposed to move in the convention of democracy and consensus and so forth. But at least this is driven by the clinical aspect. And there's the added incentive, of course, that for the clinicians who are involved in this is certainly prestigious, and it involves papers and so forth, landslide and advancement in rank. And so there are positive things of it, right? But it's still two-edged sword for sure. Anyway, it's kind of a little glimpse at the future of where we can go on the positive side. Okay, so that's what I have today. Now, I think next week, we still have a class in 2 weeks. There you go, 2 weeks. Okay, yeah, because the spring break is coming up. All right. Good. So we'll begin with the basic biology of end-of-life issues next Saturday, and then go from there. Questions, comments?
Next Saturday.
We're going to begin, and then there's a break, just then coincide with the, I think we still have all told about 7. We have a total of 14, I believe. This is the 7th. Yeah, today's the 7th lecture, and we have a total of 12. April, yeah, we're going till May, right? First week, 14, yeah.
Yes,
May 4th. Yes, we have a total of 14 lectures, which will take us to May 4th. So we're going to have 7 lectures on end-of-life issues. And then the following Saturday, May 4th, ends this semester, and May 11th, we start the next one.
Yes.
So it's going to be fast. It's going to be fast. But then, so that's the downside, is that we keep going. The upside is that we'll end at the end of June, and then hopefully we'll all have July and about half of August. Okay? And then when you come back, it's just the healthcare bioethics, because...
So we have statistics in the zone.
Yes, in addition to the environment. That's the 2. Yeah, but they'll run. Oh, thank you very much, because I forgot to ask you last time. It's the one course that I don't teach in the program, and I need to know what kind of statistical background you have, because we have a new professor teaching it, and she wanted to get a feel for the statistics for the level of the class. I told her it's going to be heterogeneous, because we have anywhere from medical doctor to a priest, and a large biologist, a nurse. So what kind of background do you have in statistics? Have you had some? In college, okay. Most of us have had some statistics in the undergraduate at the bachelor level, which I assume everybody had, right? And that may have happened a while back. How about
Long time ago.
a long time ago, long ago and far away,
What do you mean?
maybe even last century, right? For me, it was last century.
Play solitaire.
Yes, okay. Statistically speaking, for probability, yeah?
I'm also married to a math nerd, so if we ever use
Okay.
these statistics discussion.
Oh, yeah, okay.
Oh, these tutors would be statistics
Oh.
in the doctorate's level.
Oh,
So,
super.
yeah.
Okay, so that's a resource potentially for, what I do is I take the course with you also, so, because I want to make sure that everybody passes and passes well, all right? So we're in this together for the long haul. And
But still on Saturdays.
no, that's the other thing. It's not on Saturdays. It will be a week evening, and we need to pin that down. But before leaving the subject of the background, has anyone taken statistics at the graduate level? Not at the graduate level. All right. Because sometimes I do have someone, I had an accountant in one of the groups, and he definitely had statistics, so he was able to bail out of that course. He could do a substitution. But if undergraduate doesn't count, it has to be graduate level. So there's no one with a stat course at the graduate level.
I think
Hema May, yeah?
I think she
Yes, yes, she mentioned.
I
in
had
fact.
some in medical school, but it was like maybe like for public health.
Well, if you want to dig it up and pass it on, I'll be happy to run it with the statistic professor and see if we can validate. But if you want to,
Me too, because
There's
there's
really, it's
there
like
may be
doesn't
a
count. Yeah,
Okay.
It's been too long.
yeah, okay, fine. Now, the other option is also that if you have some kind of graduate course in statistic or probability or something like that, we can try to get it substituted, and you can always come to the class and participate, and at least you don't have the pressure of making a grade, all right? It's up to you, I leave it up to you, but it's also because she's not available on the weekends, so it would have to be week evening. And the typical session here is from 6 to 10 p.m. or 6 to 9 or something like that, because being blended, she can also give work to do at home, which counts toward the contact hours.
Would we be able to do it online? Like
Um,
watch the class actively?
So it will be blended, meaning that we're going to have the Zoom going on, and you don't have to come physically, but you will log in like that and follow the class, follow the lecture. Yes. On weekdays, yes, it is harder.
It will be
So
only one day.
it will be one day for about 3 hours, just like this. And the Saturday with me for the environmental. So you're gonna have the two. So you have the two classes.
Yes.
So you have two. It's really an extra class, okay? So typically Tuesday, Wednesday, or Thursday. Any preference of those 3 days? 6 to 9, more or less.
Thursdays are horrible.
Service or bad. Thursdays you're only open.
Usually,
Thursday.
I would probably be doing it from home regardless.
From home,
because
yeah.
It would be too far for me.
Right.
On a weekday.
On the weekday, yeah, the traffic is an issue. It's a big issue.
No, it's not so much the communities, but it's the day that I have to go to 3 different facilities presentation.
Wow. Okay, so it's a long day. Mhm.
So every day is busy. So I travel all day, every day to bunch of different
Everyone.
locations.
Ready? You two also, right?
Well, yeah, right.
So all right, so Thursday, we can at least rank them and see. We have to see also her availability, but Thursday would be the least one. How about between Tuesday and Wednesday? Any particular preference?
For me, it's the same.
The same?
It's far.
So either Tuesday or Wednesday would be a priority.
What's going to be the format of the class? Is she going to give us homework?
More or less the
instead?
same, but she'll have some homework. She'll have some homework. She's very interested in you doing practice.
Right.
Practice, all right. And but she'll be basically the same way. I think she also wants to have a live board where she can write, which can be done
Yeah.
also. And so she may have some PowerPoint and then some writing, but it will also be filmed like this and transmitted through Zoom, right? We'll figure out the logistics, but you'll be able to connect remotely to the class. And if you miss a particular session, then you'll have to go online and see the video just like we do here, okay?
And both classes are running parallel for the entire.
Yes, for the entire 8 weeks. Exactly. So it will be the same.
Because if Wednesday we have to turn in the homework, I'm environmental.
Right.
So
It gives you an extra day. It's
so
not.
then
It doesn't.
I think
It does. So Tuesday would be. Yeah. I mean, we could.
Maybe like every
Or
other
even-
one, I have to just watch the video because I
Yeah.
have to work on Tuesday evenings, but not every. So it still works better. than
Tuesday would be the best day.
Tuesday better. And for even on that argument, maybe even Monday, if she's available on Monday, would Monday be even better? Or about the same? Monday even better, right? In other words, there's no definite commitment that any of you have specifically on Mondays. So Monday or Tuesday would be best.
Tuesday works better.
I think that
Yeah.
the Thursday is the one that.
Yeah.
Thursday,
But
see?
Tuesday is the best because that's my choice working
Yeah.
day.
And I presume Friday also. Friday is even worse, no?
Fridays- No, and Fridays is good.
Yeah, from.
I don't know about Friday.
Yeah.
Maybe
Friday,
that's because I'm so tired. By the end
Friday
of the
may
week,
also
I'm
be.
exhausted.
Yeah.
Because I still have to work when I come home. I have to document. I have to call so many bereavement clients. There's no break.
There is. It's just ongoing.
My
It
work
really is.
already.
You're the first person I need that makes Friday.
I look forward to sitting in class. Like it's like nice. I'm
No,
sitting
right,
down.
exactly, because you're away from work.
Yes, it's wonderful.
Right. I get it. Yeah. Okay, well,
I prefer to come to class without no distractions. Really.
yes.
Stay home sometimes somebody might interrupt you.
Yeah, that's what I
Exactly.
I know you saw my cat on the video last
Yes,
week.
yeah.
That's my distraction.
Yeah.
is the cat.
Yes. Sometimes there is background. That's why I say you can mute
Yeah,
the
that's
sound and you can
That's why
minimize
it's good.
the.
There's
the
also
video.
another cat that likes to stand behind me.
Oh yes, yeah.
And he'll watch me the whole time. So
True.
I'm like, that's so weird. Maybe he's super creepy. Like if I send him to work and so he sits behind me and watches me.
So has that cat registered for the course?
Yeah.
Okay, we'll give him audit status.
For me, it's the same. This is my me time when I'm in class.
Okay,
It's my
yeah.
me time.
I
No,
know.
my mother-
And there's nothing like being live, really. There's nothing like being live face to
Yeah,
face. It's the most efficient teaching learning experience.
a little bit.
Way
I
south,
work
yeah.
at in Compass. We have hospital.
Oh, you do?
Yeah.
I work there. That's the hospital.
So that's quite a commute. Okay, so I have a feel for the best days, and let me hear from Monica and Hema, and then I'll present it to
This will be in
the
the summer.
doctor.
I go all
Yeah.
May and June.
May and June.
May and June. The second half of May, as soon as we finish here, second half of May and all of June. We'll start at the end of June.
Who are you working with?
Okay, I'm going to close this down so we can start the, let's see, stop recording. Thanks again. See you next time. Bye-bye.